Hey there, science fans. Today we're diving into the fascinating world of bacteria. Bacteria are tiny, single-celled organisms that are found everywhere on Earth, from the soil beneath our feet to the air we breathe. They live in water, on our skin, and even inside our bodies. Bacteria are prokaryotes, which means they lack a nucleus, a special compartment that holds DNA in other types of cells. Don't let their small size fool you, though. These microscopic marvels play a huge role in our lives and the planet's ecosystems. They're involved in everything from decomposing organic matter to helping us digest food. Understanding bacteria is crucial for appreciating the intricate web of life on Earth. So buckle up, explorers, as we embark on an exciting journey into the realm of these microscopic wonders. Bacteria are incredibly diverse, and they come in a multitude of shapes and sizes. One of the primary ways scientists differentiate between various types of bacteria is by examining their shapes under a microscope. Imagine peering through a powerful microscope. You'd be amazed to see these tiny organisms displaying a variety of forms, each with its own unique characteristics. The three main shapes that bacteria can take are spherical, rod-shaped, and spiral-shaped. Each of these shapes has its own name and specific features. The spherical bacteria are known as Cocci. These bacteria are round and can often be seen in clusters or chains resembling tiny beads or pearls. When Cocci bacteria group together, they can form different patterns. For instance, some may appear in grape-like clusters, while others form long chains. A well-known example of Cocci bacteria is Streptococcus pneumoniae, which is responsible for causing ear infections and even pneumonia in humans. Next, we have the rod-shaped bacteria, which are called Bacilli. These bacteria look like tiny cylindrical pills or rods. One of the most famous examples of bacilli is Escherichia coli, commonly known as E. coli. This bacterium is found in the intestines of humans and animals and plays a crucial role in digestion. Finally, there are the spiral-shaped bacteria known as Spirilla. These bacteria have a unique twisted shape that resembles a corkscrew or a spiral. Spirilla are often found in aquatic environments where their spiral shape helps them move through water with ease. An example of Spirilla is Leptospira, which can cause a disease called leptospirosis. This disease can affect both humans and animals and is often transmitted through contaminated water. So, the next time you think about bacteria, remember that they come in these fascinating shapes. Each shape not only helps in identifying the bacteria, but also gives us clues about their behavior and habitat. The incredible diversity of these microscopic organisms is truly astounding, and their shapes are just one of the many ways they exhibit this diversity. From spherical Cocci to rod-shaped bacilli and spiral-shaped spirilla, bacteria are a testament to the wonders of the microscopic world. Now let's talk about a cool technique scientists use to tell different types of bacteria apart, the Gram stain. This method is a cornerstone in microbiology and has been used for over a century to classify bacteria into two major groups. This method, named after its inventor Hans Christian Gram, is like giving bacteria a special bath that colors them differently based on their cell walls. Gram developed this technique in 1884, and it has since become a fundamental tool in bacterial identification and classification. The Gram stain involves a series of dyes that stain bacteria either purple or pink. The process starts with applying a primary stain called crystal violet, followed by a mordant, usually iodine, which helps fix the dye to the bacterial cell wall. Bacteria that turn purple are called Gram-positive. This is because their thick cell walls retain the crystal violet dye even after a decolorizing agent such as alcohol or acetone is applied, while those that turn pink are Gram-negative. These bacteria lose the crystal violet dye during the decolorization step and are then counterstained with a red or pink dye such as safranin. This color difference reflects fundamental differences in their cell wall structure. Gram-positive bacteria have a thick peptidoglycan layer, while Gram-negative bacteria have a thinner peptidoglycan layer and an additional outer membrane. Gram-positive bacteria, like Staphylococcus aureus, have a thick peptidoglycan layer in their cell walls which retains the purple dye. This thick layer provides structural support and protection. Gram-negative bacteria, like Escherichia coli, have a thinner peptidoglycan layer and an outer membrane that allows the purple dye to wash away, leaving them pink. This outer membrane can also act as a barrier to certain antibiotics. Knowing whether a bacterium is gram-positive or gram-negative is super important for doctors. This information can guide them in diagnosing infections and determining the most effective treatment plan. It helps them choose the right antibiotics to fight infections. Some antibiotics are more effective against gram-positive bacteria, while others target gram-negative bacteria. It's like knowing which key unlocks a door. By understanding the gram stain results, doctors can make informed decisions that lead to better patient outcomes and more effective treatments. Just like us, bacteria have different relationships with oxygen. These relationships are crucial for their survival and dictate where they can live and thrive. Some bacteria need oxygen to survive, just like we need air to breathe. These bacteria are known as aerobic bacteria, and they play a vital role in many ecosystems. Others can't stand oxygen and will actually die if exposed to it. These are the anaerobic bacteria, which have evolved to live in environments completely devoid of oxygen. And then there are those that can switch it up, using oxygen when it's around but surviving without it when it's not. These versatile bacteria are known as facultative anaerobes. Bacteria that love oxygen are called aerobes. They thrive in oxygen-rich environments, such as the surface of the soil or in our lungs. They need it to produce energy from their food. This process, known as aerobic respiration, is highly efficient and allows these bacteria to grow and reproduce rapidly. Think of them as the athletes of the bacterial world. They are constantly active and full of energy thanks to their ability to harness oxygen. Mycobacterium tuberculosis, which causes tuberculosis, is an example of an aerobe. This bacterium thrives in the oxygen-rich environment of the human lungs. Bacteria that can't tolerate oxygen are called anaerobes. These bacteria have adapted to live in places where oxygen is absent, such as deep underwater or in the intestines of animals. Clostridium botulinum, which causes botulism, is a well-known anaerobe. This bacterium produces a potent toxin in oxygen-free environments, making it a serious health threat. And then we have the facultative anaerobes, the bacteria that can switch between aerobic and anaerobic respiration depending on the availability of oxygen. This adaptability allows them to survive in a wide range of environments. They're the adaptable ones, like Escherichia coli, which can survive in both the presence and absence of oxygen. This flexibility makes them incredibly resilient and capable of thriving in diverse conditions. Isn't it fascinating how these tiny organisms have evolved such diverse ways to obtain energy? From using oxygen to avoiding it entirely, bacteria have developed a variety of strategies to meet their energy needs. It's yet another example of the remarkable adaptability of life. Bacteria, despite their small size, demonstrate an incredible ability to survive and thrive in almost any environment, showcasing the resilience and versatility of life on Earth.